Okay, so welcome to week 3, and I wanted to go through a quick review of week 2 and just kind of remind you and kind of show you what happens in DNA replication. And this is a process that's going to be completely different than what we're going to talk about in week 3, but I just wanted to review it. Okay, so remember the enzyme that comes in and reads and makes new DNA, that's called elongation. The enzyme that does that is called DNA polymerase. And this one specifically that we're looking at is DNA polymerase 3. All right. What direction does this travel? DNA polymerase can only travel from the 5' end of DNA to the 3' end. Also, what it does, it reads the base pair and adds the complementary base to the new DNA strand. Okay, so if it reads a C, it's going to pull in a G and attach that, and then it's going to read another C and it's going to attach a G. What would be next here? This is reading a G. What would be the complementary base pair for G? The answer is C. And then the next one it would read would be A, so it would bring in T, and then T, it would bring in A. So that's just a quick review of what we talked about with DNA synthesis or DNA replication. So the next process we're going to talk about is RNA synthesis, and this is often called transcription. Okay, transcription is the making of mRNA. So what I did is I wanted to go ahead and just look at what is mRNA and how is it different from DNA. Well, if you notice, mRNA, M stands for messenger. All right, so this RNA is going to carry a message somewhere in the cell, and we'll learn about that in just a minute. So mRNA is a little bit different than DNA. One thing you need to remember about mRNA is single-stranded. So if you look at this picture, it shows a single strand of an RNA molecule, and this is going to be messenger RNA. Okay? I think you can go ahead and point out a really big difference between this, just not only that it's single-stranded, but there's a different base pair here. Can you see what it is? All right, it is the base pair uracil, okay, and we represent that with the letter U. What base pair do you not see in this RNA? You do not see thymine. Thymine will never be found in an mRNA or any RNA molecule. Okay, so you can think of it as U takes the place of T. So if we were looking at the complementary base pair for these, A would bond to T, U would bond to A, U would bond to A, G would bond to C, C would bond to G, C, G, A, T, G, C. Okay, so on an mRNA molecule, you can only have U's, you cannot have any T's. Okay, so I think that's a good review and overview of what is mRNA. Okay, we'll learn a little bit more specifically about what mRNA is in a minute. Okay, so the process of transcription is almost identical to DNA replication. So what we have to do is we have to actually read the DNA strand just like DNA polymerase did, but in this case we're making mRNA, so we're going to use a different enzyme or a different protein, and this protein is called RNA polymerase. So RNA polymerase reads a DNA strand, it adds the complementary base pairs into a single mRNA molecule. So let's see if we can figure out what this RNA polymerase just did. So remember, these enzymes, the polymerases, they can only read from the 5' to the 3' end. So this end would be the starting point, and this end would be the finishing point. So here is RNA polymerase, and it started here with C. Okay, it bonded with G, T with A, G with C, G with C, C with G. This is where it was different. A bonds with U. Remember, RNA is single-stranded, okay, and RNA does not have thymine. So A bonds with U instead of T, A bonds with U, and this T, it recognizes this T and it bonds and it makes an A. Okay, so I think that's pretty straightforward. Okay, some things about mRNA that you need to know. All right, just remember, mRNA is single-stranded. mRNA has uracil instead of thymine. And if you look, I've circled 3 of the mRNA base pairs. Okay, in mRNA, every third or every 3 mRNA base pairs represent something. Okay, and we have a term for that. So the 3 nucleotide sequence in mRNA, that is called a codon. In a later slide, we're going to look at these codons and we're going to see that they're important when we talk about protein synthesis. Okay, so this would be a codon, that would be a codon, and that would be a codon. Okay, so just make sure you kind of understand that as far as mRNA. Okay, so make sure you go back and you review this video if you have any questions. Make sure you go back and you take good notes. Write down any questions that you have and you can email those to me. And I will see you next week.